Hi, everyone. Thank you very much for joining us today. We are here for the webinar, How to Use AI for Classifying Cell Types and Phenotypes in the Cloud, the Live Dead Assay. And we have a featured speaker today who is Dr. Ilya Goldberg. He was recently appointed as our Chief Science Officer. And today's webinar is presented by our company, ViQi. My name is Julie. I'm on the marketing side here, and I am going to kick us off. So let's get introducing Dr. Goldberg. Ilya is here and he'll be speaking to you guys today. He has a long career that lies at the intersection of biology, imaging, and AI. He has done things like co-founding a company that developed the first medical device to receive regulatory clearance for using an AI to predict malignancy in lung nodules in CT exams. He led a research group at NIH, National Institute on Aging, which developed machine learning software for image processing in biology and medicine. While at MIT, he co-founded the OME project, which is still used for imaging infrastructure in large image repositories today. And if that isn't enough, he has over 60 peer-reviewed scientific articles from his time at Johns Hopkins, Harvard, MIT, and NIH in subjects like molecular and cell biology, pattern recognition, image informatics, and the basic biology of aging. So, Ilya, I'm going to hand it off to you.
Hi, everyone. This is an example of how you would train a very simple AI, which is a binary classifier. And there's a lot of examples of binary classification problems. So we kind of put it in the title, the live dead assay is very sort of common assay to do in cell biology or just biology in general with imaging. But you can generalize that. I mean, a lot of things have positive and negative controls. Well, positive and negative controls are a perfect thing to train an AI on. And it'll tell you, does your thing look more like a positive control or more like your negative control? And an equivalent to that is treated and untreated samples. In clinical work, we have a lot of case control studies. I mean, a lot of medicine is based on case control studies, and that's essentially a binary classification, like benign, malignant. So in the example screenshots that follow, we're going to tag areas of an image and with labels, and then we can tag, or alternatively, we can tag whole images with a particular class. So let's start with the next screenshot. So this is a screenshot from ViQi's platform, and you can do this in a browser where you're presented with an image. Here we're just looking at bright field, but you can see that we have other labels in here that we can turn on and off to allow us to manually by eye label these cells, because we know that they're one thing versus another thing based on our knowledge of the system. And so this is our input into the AI for what we wanted to do eventually and label cells for us, right? So in the next slide, we can alternatively have a conventional image processing algorithm go and segment out all the cells for us, and then we would label them afterwards. And here, the colors are just showing us how the segmentation algorithm is separating cells. So cells that are close to each other, potentially touching, would have different colors assigned to them based on the cell ID, if you will. So those can be labeled also as an alternative path and sent to the AI. And in the next slide, and see, well, what is it that we're actually sending to the AI? So that when you click on cells, or the segmentation algorithm identifies them for you, you can extract a lot of standard sort of descriptors of these cells. There's the image mask, that the individual pixels that comprise the cell. And you can get various kinds of textures or the perimeter shape or form factors, etc. And you can, those are natural sort of descriptors, numerical descriptors of what these cells are. And so, these are all in all these different columns here. And there, of course, the cell has a representation that we saw in the previous images in the different fluorescence channels. So there's descriptors of the bright field channel and separately descriptors in all the other channels that you have for the same cell. And they could be the same descriptor, they just apply to the other channels. So what you end up with is this feature table. I think this one is 60 columns wide or so. We're looking at just the a piece of it. Also, you see here that's highlighted towards the middle is the class column. So the class column is the other very important input into the AI. And this is called different things. Typically, either the class or the label is also sometimes used as the name for this. This is your target. What are you training the AI to do? You're training it to predict the class, right? And you're training, because this is supervised learning, to do the training, you're giving it which set of columns for a given row correspond to a particular class that you've labeled, right? And so those together are given to the AI. The AIs, the feature-based AIs, they don't care what this numerical content is. This could be categories or floating point numbers or whatever you want, and it doesn't care how many columns you have either. I mean, so really the AI's task at this point is has nothing to do with images. It's simply using all of the columns here, but all of the columns other than the class, to predict what the class column would be. And you can actually set the AI to be that general. So given a table, here's the class column, and use all the other columns to predict the class. And AI systems that are fairly automated would go ahead and do that. So that's essentially what you're doing when you're training a feature-based AI. Of course, in deep learning, you would just feed it the pixel mask, and it would figure out all of this numerical content internally. So next, we'll look at, in our case, deep learning AIs are almost there for push button AI. With feature-based AIs, they can't, because the number of parameters that you have is fairly small. You can automate the whole thing and have it run through multiple different algorithms and choose them for you automatically based on parameter searches. And so you can have essentially a very, very simplified way of training the AI. And so you have input data, which is your collection of images that all have the same set of columns and some labels for what you decided what was what. Then you have the other parameters, so that's the input data. Then you have the other parameters, which really just let you say, are you interested in labels on particular objects? So what kind of cell is this in a field of cells? Or are you interested in labels attached to images? So this image is from a well that was treated, and this image is from a well that wasn't treated. So you're basically selecting what tag you're going to use for the training. And that's it. The AI should really do everything else. So you click the run button, and then you get a result, which is in the next slide. that would be, in this case, a labeling of cells by class, right? So now the color coding is by what the AI predicted to be the class. And again, you need to evaluate how well the AI did. Since you manually trained it, you have to have you should have at least a chance to evaluate visually what the AI did and get your impression of whether it's doing a good job or not. But there's obviously numerical ways of checking how well the AI did, and that will go in the next set of slides. So the first thing is a training curve, and that's to answer the question of, is there enough data? And so you can see an example of really successful training on the left, where so notice the scale, first of all, that it goes from 90 to 100. So we're looking at the very top of the scale for performance. So this is the scale. On the Y, we're looking at accuracy, essentially. So 100% would be a fully accurate classifier, right? And on the bottom, we're looking at the fraction of the samples that we used to get that level of performance. And so you can see as you increase the samples, you get better performance. And then you have kind of a plateau, or at least you have a region of seriously diminishing returns, and it plateaus out at a fairly high level. So we get, with 60 or so % of the samples, we are essentially topped out at just short of 98% accuracy. So that's really good. In contrast, on the right, you have a situation where you have many samples, and you're kind of reached the diminishing return area of AI training, and it's still not great. It's at 60%. And so the answer here is that, yeah, you have enough data, but it's not, for whatever reason, it's not enough to train an AI. The AI is not performing well. Whereas on the left, you've successfully trained your AI, and now you can test it on real unknowns, or actually you want to test it on a withheld data set first. The other thing to look here and in the coming slides is the error bars. So you want to make sure that each one of these dots is a separate AI that was trained on the indicated number of samples. And you want to make sure that each time you train an AI, it's based on a subset of your training data, and then it uses the rest of your training data to check it. And so you want to make sure that the sampling is such that you don't have a lot of variability. It looks like a lot of variability with the one on the left, but it's actually very small because the scale is extended. So that's the training curve. Next is parameter sensitivity. So the same idea here, you're sweeping across different kinds of parameters. You want to make sure here that your performance is good, obviously, but that you're not in a sharp little peak, because if you move even a little bit off of a sharp peak, right, your performance would suffer. So that means if you change your parameter by a little bit, you would cause it to your AI to not perform as well. And so here you can see these are sort of all broad regions, and this is support vector machine parameters, and there's only 2 of them. There's actually 3 in the whole 3 parameters for the whole AI training. And so this is telling you that this is a nice stable sort of AI that we've trained here. And next we have the confusion matrix. So this here we're using a different example. This is lilies, I believe. We're classifying lilies based on some input data. And so a confusion matrix, the way you read it is that the known class. So at the top here, Setosa, right, is the known class. And then each column is the predicted class. So Setosa is being predicted as Setosa 13 times out of 13, right? So great. And on the left, we're looking at percentages. I mean, on the right, we're looking at percentages. On the left, we're looking at individual calls. And they're 100% correct. In contrast, for varies color, we have some error with Virginica, right? And so it gets them mostly right, but there's some confusion between versicolor or Virginica, right? That's why this is called a confusion matrix, because it lets you identify classes that are confused with other classes. And this poses some questions about how you might want to restructure your AI in order to improve the situation if it needs to be improved. So let's keep going to overtraining. So I talked a little bit about overtraining and how you have to really be careful about it. The message here is you really should withhold a set of data for training once you have a candidate AI that's been trained, right, to test on your withheld data. So in other words, you know all the answers, but the AI has never seen that data before, and you really want to evaluate your AI based on that, rather than its internal checks that it will report to you, because those checks are done during training and not with a completely withheld set. And this goes into a little bit of difference in how we talk about validation and testing. So in the AI world, so that's machine learning, AI, deep learning, data science, the validation sets, people typically talk about these internal sets that the AI uses to see how well it's doing internally, right? And the test set is this withheld set that the AI never sees during training that you use to determine how well the final AI is performing. This is actually very different in how validation and testing is used in the regulatory field and possibly in common use also. Validation there is that final set that you use to validate the final AI model, right? The test sets are the ones that you use for internal testing while you're developing your AI. So be very careful about when people talk about validation and testing, what they're talking about, because they mean different things in these fields. So let's go next slide. I wanted to talk a little bit about bias, and that will be the last part of this webinar. So the most common source of bias is disproportionate class sizes. And that's where if you have a training set, 90% of it is class A and 10% of it is class B, a predictor can, if it always guessed class A, no matter what, right, it would be 90% accurate, right? So you have to be, when you're dealing with disproportionate class sizes, you have to be very careful about how your AI is tested and what your accuracy figure actually means. And you have to be careful about how it's trained, because some AIs will actually train themselves to the point and say, well, I always win by guessing class A in this case, so that's what I'm going to do. So you don't want the AI to do this. You want to force it into making an actual decision. And so really there are ways of compensating for this, but if you can, the best way to do this is to force the classes to be equal sizes. Then the other last bit of, we'll talk about bias is on the next slide. These are, your data is often skewed simply, and even if you have equal class sizes, they may be different things that are misrepresented that you haven't accounted for. That's sort of the typical bias that you might have in your training data. And it's not enough to just say, well, I have random training data, so it's not biased, because the bias could still be there. There are a couple of things you can actually do. It's not hopeless. So first of all, you can measure the bias. You can sort of say, look, I have a bias variable here. Images were collected by 2 different postdocs. And, can the AI distinguish between the 2 postdocs? So you make, instead of treated and untreated, you say, Bob and Margaret collected these images. Can the AI distinguish them? If the AI comes back and says, no, I can't, so it has bad performance, then it's not sensitive to bias, and so everything's fine, right? On the other hand, if it comes back and says, yes, I can distinguish them, right, then you can still do some things about that. So you can confuse the AI with regards to the bias. So in other words, you have an even distribution between your 2 postdocs in each class. And so the AI has nothing to latch onto to train itself to just recognize one of your postdocs. And there are other ways of systematically training out this bias, which is kind of more advanced. We're not going to get into, but it is possible to train an AI to not recognize certain bias variables. And I think we're done. I think we have a parting thought here, which is that the performance of your AI ultimately is completely dependent on how accurate your class labels are, and the degree to which your training sets represent reality. And the important thing to keep track of here is that we are dealing, in fact, with tools that can see and detect patterns much better than we can. And so, first of all, when you train these AIs, you don't want to necessarily rely on a single person's idea of what is in this image if you're using image content as your training basis. You might want to get multiple reads or get a consensus read to get a more accurate class label. And of course, you have to be absolutely conscious about bias, because these are kind of black boxes. And so they could be latching on and learning from things that you really didn't intend.